Welcome to Lecture 5C, the third and final lecture for the Week 5 B-cell module of Applied Immunology. Today we will apply our understanding of B-cell development and activation towards understanding the functions of B-cells and their secreted antibodies in promoting adaptive immune responses. We will talk about the different classes or isotypes of antibodies and how each class is structured and distributed amongst tissues throughout the body. In the second half of today's talk, we will go over the effector functions of antibodies, and then tie everything together by discussing how certain isotypes optimally promote pathogen-specific adaptive immune responses. Lastly, we will briefly discuss how memory B cells are established in order to promote the long-term immunological memory that is a key characteristic of the adaptive immune system. Please note that the midterm exam you have coming up in Week 6 will cover material from Weeks 1 through 5 in this course. So today's Lecture 5C is the last lecture of material that you will be tested on for the midterm. By this point in the course, you should already know that the primary function of B cells who have undergone activation in response to direct antigen binding to the BCR is to generate soluble forms of immunoglobulin called antibodies. Antibody-mediated immunity is often referred to as humoral immunity or the humoral immune response. Today, we will cover how antibodies function to promote adaptive immune defense against infection, but let's first quickly recap some of the different types of B cells that can produce antibodies and the properties of the antibodies that they produce. In Lecture 5B, we learned about mechanisms such as somatic hypermutation and class switching that are engaged during B cell activation, which can improve the quality of these antibodies. But aside from the affinity and isotype of an antibody, the B cells that produce these molecules also differ with respect to the magnitude of the antibody response, as well as the timing of antibody generation. It's easiest to compare these responses when looking at this hypothetical graph, which plots the relative amount of antibodies circulating in serum on the y-axis versus the time post-infection on the x-axis. In the last lecture, we learned that some mature B cells can bind to polyvalent antigens called thymus-independent antigens, or TI antigens. These antigens can trigger B cell activation and differentiation into plasma cells that secrete antibodies with the IgM isotype, which is the default isotype that's expressed by mature naive B cells before they encounter antigen. These IgM antibodies provide an important level of protection very rapidly within the first few days of infection, but their serum levels decrease after about a week or so, as you can see in the purple curve on this graph. We also know that there are B cells that bind to thymus-dependent antigens, or TD antigens, which means that they need to receive T cell help from T follicular helper cells, or TFH cells, in the secondary lymphoid organs. T cell help allows for the activation and proliferation of these B cells, which then, in the absence of further TFH interactions, can immediately differentiate into either plasmablasts or plasma cells. Again, plasmablasts form a structure called the primary focus, where they proliferate rapidly and produce larger quantities of antibodies for a week before they start to die off. Plasma cells can be long-lived in comparison, in which case they can migrate to the bone marrow to form a memory population. Both plasmablasts and plasma cells responding to TD antigens secrete IgM antibodies early on in infection, although due to their interactions with TFH cells, this antibody response is a bit delayed relative to the T-independent antigen response, and this is represented by the blue curve on the graph. The immediate TD response does produce a slightly higher level of antibodies due to the activity of plasmablasts that are proliferating in the primary focus. Lastly, we found out that some B cells responding to thymus-dependent antigens maintain contact with TFH cells in the B cell follicle, and these form a structure called germinal centers. These B cells undergo repeated iterations of somatic hypermutation that enables the process of affinity maturation, and this is where the immunoglobulin variable region's affinity to antigen is selectively increased over time. Germinal center reactions are also where class switch recombination occurs, and antibodies can undergo class switching to different isotypes, such as IgA in this example. This process therefore yields plasma cells with higher quality immunoglobulin specificity, as well as isotype matching to the type of infection. Because germinal center reactions depend on these repeated cycles of somatic hypermutation, germinal center antibodies take considerably longer to produce compared to the other two sources. As you can see on the graph, the orange curve representing the IgA germinal center antibodies doesn't start to come up until about a couple of weeks following infection. However, antibodies from GC-derived plasma cells are produced at much higher amounts that are maintained in serum for longer time periods compared to the immediate IgM isotypes. Obviously, the production of large amounts of high-affinity antibodies from plasma cells that arose from the germinal center makes these higher quality antibodies than the IgM isotypes that are produced earlier on during infection. However, in addition to their affinity, how exactly do different constant regions or isotypes influence the downstream immune responses that are sort of engaged in response to these antibodies? Now, there are 5 major antibody classes or isotypes that are found in mammals such as mice and humans, although only 4 are thought to have any function. We first heard about these during our week 4 lecture on lymphocyte antigen receptor structure and generation, when studying the different constant or C gene segments that can be used to make the immunoglobulin heavy chain. The organization of the genetic locus encoding these C segments is shown in the bottom row of this figure for both mice and humans. Because a single B cell clone can proliferate into many different daughter cells with the same immunoglobulin variable region specificity, through class switching of C gene segments, it is possible that one individual could contain B cell clones that have the same antigen specificity that's basically matched onto different antibody isotypes. Again, the process of class switch recombination is controlled through cytokine detection by B cells, though we will talk about how those cytokines tailor antibody responses later on in this lecture. Antibody isotypes differ with respect to a number of structural components, including the number of domains that make up the constant region, the number and location of disulfide bonds that hold the immunoglobulin molecule together, and the distribution of carbohydrate groups throughout the constant region. More importantly, these structural differences translate into functional and tissue distribution differences between different isotypes. Let's go through each one of these just one by one to start to cover these differences. First, by examining their structure and distribution amongst different organs and tissues. We can start with the IgM and IgD isotypes that we've already encountered during B cell development. We know that at the end of B cell development and selection, all mature naive B cells express their BCR using both IgM and IgD isotypes. Very little IgD antibodies are secreted at any point in an immune response, and the exact function of this isotype is unknown, so we're not going to really talk about it much from here on out. IgM is the first isotype used for antibodies secreted by activated B cells, which from the previous slide we know is extremely important for offering immediate protection early on in infection. However, because IgM antibodies haven't undergone any form of germinal center reactions, these form relatively low affinity antibodies compared to other isotypes. Note that in a human adult, the IgM isotype only makes up about 10% of the circulating antibodies that can be isolated from serum. An important structural aspect of IgM is that it assembles these multimeric structures that are made up of 5 IgM molecules which join at their constant region. And this multimeric oligomer is called an IgM pentamer. And when this was first discovered, it was referred to as a macroglobulin, which is actually what the M in IgM stands for. The constant regions of an IgM pentamer are held together by a small polypeptide called the J chain, shown here in yellow. And this forms an overall star-like shape for the entire complex. This multimeric structure is important with respect to 2 properties of antibodies that have kind of annoyingly similar names, and these are called affinity and avidity. We know that affinity refers to the strength of the binding interaction between a single variable region with a single cognate antigen, and that IgM antibodies, again, are low affinity because they haven't entered a germinal center reaction. Now, avidity, on the other hand, refers to the overall number of antigen binding sites that are available in a given molecule or complex. Now, because each antibody in the IgM pentamer has 2 variable regions, the pentamer as an entire complex therefore has 10 available binding sites. This means that IgM pentamers are high avidity complexes, which can compensate for the low affinity of the individual antibody monomers. So keep in mind that multimeric antibody complexes do not influence antibody affinity, but they do change the avidity of a given isotype. Also, the larger size of the IgM pentamer means that the structure is difficult to diffuse or transport amongst tissues. So it is primarily found in the blood and to a lesser extent the lymph. The first type of class-switched antibodies that we will cover is IgG, encoded by different C-gamma gene segments. Unlike the other isotypes, which are encoded by only one gene segment, IgG comes in 4 different subclasses, and in humans these are IgG 1, 2, 3, and 4. Unlike IgM, IgG antibodies are always monomeric, so this is a low avidity antibody isotype. However, the smaller antibody size and the lack of multimeric structures means that IgG is a smaller protein, and this allows it to diffuse into tissues. IgG is therefore found in extracellular fluid throughout the entire body, as well as in the blood, where it is the most abundant antibody isotype. Both IgG and another isotype called IgA are the most abundant antibody isotypes in the body. In addition to diffusion, IgG can be transported across the placenta by a specialized neonatal form of an Fc receptor. We will discuss the functions of Fc receptors near the end of today's lecture. So maternal IgG antibodies therefore can be transferred from mother to fetus during pregnancy, or while the fetus is in utero, and this is an important form of immune protection in newborns against pathogens that are found in their environment. Because class switching usually occurs in germinal centers, IgG antibodies have typically also undergone somatic hypermutation. So compared to IgM, IgG antibodies are relatively high affinity. Because they are secreted as low avidity monomers, this increased affinity of IgG antibodies is important for their efficacy. As a general pattern, you'll see that class-switched antibodies resulting from germinal center reactions have high affinity, and this means that they don't need to form large multimeric complexes for their activity the way that IgM does. The next category of class-switched isotype is IgE. As with IgG, IgE only exists in monomeric forms, which means that it is smaller and capable of diffusing into tissues. However, IgE is a relatively low abundance isotype and is found in the blood and interstitial fluid of epithelial tissues such as the skin and lung. Like most class-switched antibodies, the specificity of IgE antibodies has undergone somatic hypermutation in order to make these high-affinity antibodies. Although IgE is a rarer form of isotype, it is very important for defense against pathogens that infect epithelial surfaces, especially parasites. This isotype is produced in response to parasitic infections, and as we will find out in the upcoming slides, other immune cells that respond to IgE antibodies are those with important functions for type 2 immunity against parasites. We will also learn in week 9 that these can be sort of inappropriately activated in the context of allergy or hypersensitivity reactions. The last antibody isotype is IgA, which again are usually high affinity antibodies resulting from germinal center reactions. IgA can be secreted in a monomeric form, but it can also form higher avidity dimers through J-chain linkages. Even in its dimeric form, IgA is smaller than pentameric IgM, and like the other class-switched isotypes, is therefore able to diffuse out of the blood and into tissues. IgA is highly enriched in secretions associated with epithelial barriers, which are called mucosal surfaces. This includes tissues like the gut, lung, and male and female reproductive tracts. IgA is therefore said to be extremely important for mucosal immunity against pathogens that enter the body through these barriers. Note that monomeric IgA is typically produced by plasma cells located in secondary lymphoid organs, while dimeric IgA, again, two IgA molecules that are linked together, is formed at mucosal surfaces such as the gut. IgA is relatively abundant and, along with IgG, makes up the predominant antibody classes that are found in human circulation. IgA is also important for maternal immunity, although unlike IgG, IgA is not transferred to the developing fetus via transplacental transport. Instead, IgA is secreted through the epithelial tissues lining the milk ducts of nursing mothers. So IgA is therefore transferred to a newborn through breast milk. This means that once a newborn begins breastfeeding, they have received both IgG antibodies from the mother during their development in utero, as well as IgA antibodies that can function in the newborn's gastrointestinal tract. These two forms of maternal antibody transfer are extremely important forms of protection for newborns who haven't yet developed adaptive immune systems to protect them from infections. This first half of today's lecture has outlined how the antibody isotypes differ with respect to the structures that they form and how this influences their distribution amongst various tissues throughout the body. All of this information is summarized in this table from the textbook, as well as this figure mapping the anatomical distribution of antibody isotypes. Note that there's a little bit of an annoying switch in color coding conventions for each isotype between the chart on the left and the figure on the right, so just try to keep them straight. We've established that the first wave of IgM antibodies are predominantly found in blood, where they form pentameric structures that restrict their diffusion into tissues. IgG antibodies diffuse throughout the extracellular fluid of all tissues and can reach a developing fetus through transplacental transport. IgA antibodies are enriched in body secretions at mucosal surfaces, such as the gut, lung, and milk ducts. And lastly, IgE antibodies are in relatively low abundance but still mediate important functions at epithelial barriers that help to protect the host from parasitic infections in particular. Also, as a side note, the IgD isotype that is expressed by naive B cells during development is secreted in very low amounts and has no known function. Now that we understand where each isotype is found throughout the body, let's move to the second half of today's talk, where we will look at an overview of the different immunological functions of antibodies and why these are such an important component of the adaptive immune system. The antibody classes differ not only in their tissue distribution, but in their ability to mediate 5 different effector functions of antibodies. We've learned about a few of these so far in this class, including the process of opsonization that allows for phagocytosis of extracellular bacteria and fungi, as well as complement deposition that is also important for defense against extracellular pathogens. We will learn about a few more functions of antibodies in this lecture, including direct neutralization of extracellular pathogens and toxins, as well as the sensitization of different innate immune cells, including NK cells, for defense against viruses, as well as mast cells for defense against parasites. This chart summarizes the efficacy of each antibody isotype at mediating each of these effector functions. As a generalization, IgM is best at activating complement, while the IgG subclasses are decent at most functions except for mast cell activation. IgA is best at neutralizing extracellular pathogens and toxins, and again, IgE predominantly functions through mast cell sensitization to promote antiparasitic type 2 immunity. We'll go through more specific examples of each of these effector functions in the upcoming slides, but please refer back to this summary chart if you find it helpful for your studies. First, some antibodies function by blocking the ingestion of harmful molecules called toxins. You can think of these small molecules as cellular poisons that bind to receptors on the host cell surface, and upon their endocytosis, toxins interfere with cellular functions or can even kill the target cell. Antibodies prevent this process if they are specific for an epitope on the toxin itself, in which case they bind to the toxin and prevent it from ever binding to the cellular receptor. Toxins are typically expressed by forms of pathogenic bacteria, and I've listed a few of these here on the right, as I'm sure that many of you have at least heard of things like tetanus, diphtheria, botulism, etc. If you've ever heard of antivenom being used to save patients that were bitten by a poisonous snake or insect, this treatment just consists of a large dose of neutralizing antibodies that are specific for whichever toxin is present in that type of venom. Antibodies can also function by blocking the entry of pathogens into cells. Now, this can occur with bacteria that use molecules called adhesins to bind to receptors on a host cell surface, and then upon endocytosis of the receptor-bacteria complex, the bacterium can set up a replicative niche in an intracellular vesicle in the host cell. If antibodies are specific for these bacterial adhesins, however, they can block receptor interactions to prevent bacterial uptake and infection. Note that a similar process can occur with viruses, which usually require binding to a receptor expressed on the plasma membrane of host cells in order to initiate viral entry as the first stage of infection. For both categories of pathogens, antibody binding inhibits infection. Because these types of antibodies inhibit the uptake of harmful molecules or pathogens, they are called neutralizing antibodies. Importantly, they function without additional activation of other immune cells. So antibody secretion alone confers protection. The best isotypes for making neutralizing antibodies are IgG and IgA. And this especially makes sense for IgA, which is secreted into mucosal surfaces like the intestinal lumen, where it is unlikely to interact with other immune cells but can still carry out important effector functions through neutralization. Second, antibodies can function by recognizing antigens that are expressed on the surface of extracellular pathogens, such as bacteria and fungi. And in this context, they can induce complement activation. If you think back to our week 2 lecture on the complement cascade, remember that the classical pathway of complement activation can be triggered by the binding of the initiation protein C1q to the surface of a microbe in one of 2 ways. So first, C1q can bind directly to C-reactive proteins, Or second, C1q can bind to antibodies that have already recognized other antigens on the surface of the microbe. This second case of antibody binding is an important mechanism for initiating complement activation. C1q can recognize two forms of antibodies, either pentameric IgM or multiple monomers of IgG that are located close together. It therefore makes sense that both IgM and IgG are best at triggering the complement cascade. In either case, C1q binding to these antibodies kicks off the enzymatic cascade of complement signaling, which leads to either CR1-mediated phagocytosis of opsonized pathogens or direct cytolytic killing through the formation of the membrane attack complex. Therefore, complement targeting by IgM or IgG isotypes can function similarly to neutralizing antibodies in that they can function effectively without the activation of other immune cells, in the case of MAC formation that occurs downstream of complement signaling. These are the only two forms of antibody functions that can act independently of other cell types, so just try to keep that in mind. The other three forms of antibody effector functions require the activation of other immune cell types, which in this context are called accessory effector cells. This allows for the antigen specificity encoded by an antibody to be linked to effector functions of innate cells that lack antigen receptors. This linking of antibody recognition to accessory effector cell function is largely accomplished by the expression of a class of receptors called Fc receptors, and these bind to the constant region of antibodies. This portion of the antibody is also referred to as the fragment crystallizable, which is the reasoning behind the Fc abbreviation. This chart summarizes the structure and binding properties of several different Fc receptors. These are usually named using an Fc followed by a Greek letter that corresponds to the constant gene segment that the receptor recognizes, and then an R for receptor and Roman numerals for 1, 2, or 3. Note that similarly to the BCR, Fc receptors can be activated in signal when they are cross-linked with several receptors that are clustered together in close proximity to one another. Don't get bogged down in the details here since I know that this is a lot of information. I mostly want you to focus on which Fc receptors recognizing certain antibody isotypes are expressed on which immune cells, because this plays an important role in terms of their downstream functions. For example, the two Fc epsilon receptors highlighted in this red rectangle bind to the constant region of IgE isotype antibodies. Fc epsilon receptors are expressed on cells involved in defense against parasites such as mast cells, eosinophils, and basophils, each of which can become activated in response to Fc receptor signaling. Since IgE is produced in response to parasitic infections, it makes sense that recognition of this isotype should be linked to effector cell types that are involved in the antiparasitic responses of type 2 immunity. This example of IgE and Fc epsilon function can be applied to the third type of antibody function, which is called mast cell sensitization. IgE antibodies that are produced by B cells in response to parasitic infections can bind to Fc epsilon receptors expressed by a type of innate immune cell called a mast cell. Now, we haven't really gotten into mast cells yet, but these are an important cell type for protection against parasitic infection. Once IgE binds to Fc receptors on the mast cell, if the IgE antibody then binds to a multivalent antigen that's expressed by a parasite, this can induce Fc receptor cross-linking. Again, cross-linking is required for Fc receptor activation, which relays a signal to the mast cell that causes a process called degranulation. This leads to the mast cell releasing a variety of inflammatory molecules that it keeps stored in intracellular organelles that are called granules. Unlike the cytotoxic granules that we've learned about with NK cells and cytotoxic T lymphocytes, mast cell granules contain strong inflammatory mediators, such as histamine. Now, when histamine is released, it induces muscle contractions at the epithelial surface that lead to things like coughing, sneezing, and vomiting, which can help expel infectious parasites. Mast cell activation through Fc epsilon receptor signaling can also lead to the production of inflammatory metabolites, as well as inflammatory cytokines like TNF-alpha. Again, these effector functions of mast cells are very important for defense against parasites, but as we will learn in week 9, these cells can also become inappropriately activated against harmless environmental antigens, and this is what drives allergy or hypersensitivity reactions. Collectively, all of these functions of mast cells promote defense against pathogens at epithelial barriers where IgE is expressed. So the only antibody isotype that can really do this is IgE. And also keep in mind that Fc epsilon receptors are expressed by other type 2 immune cells, such as eosinophils and basophils, which can execute similar effector functions as mast cells. These functions by type 2 immune cells are very important for attacking parasites, which tend to be larger pathogens that can't be as easily killed through things like complement signaling or phagocytosis by macrophages. Another accessory cell type that can be activated by antibodies is the natural killer, or NK cell. Back in week 3, we learned how NK cells can become activated in response to signs of missing self or altered self, which are typically induced by viral infections. However, NK cells can also become activated through Fc receptor-mediated recognition of antibody-coated cells, and this is a process called antibody-dependent cell-mediated cytotoxicity, or thankfully just ADCC for short. Some host cells that are infected by intracellular pathogens, such as a virus, can express foreign antigens on their surface, which are then recognized and bound by IgG antibodies. An NK cell then recognizes antibody-coated host cells through its Fc gamma receptor, which is alternatively called CD16. If enough antibody is present to induce Fc gamma receptor cross-linking, it signals to activate the NK cell and trigger the release of its cytotoxic granules, which in turn cause the target cell to die by apoptosis. Take note that NK cells can therefore kill virally infected cells, both through the binding of activating receptors like NKG2D, which we learned about earlier in this course, as well as through recognition of IgG antibodies through the Fc gamma receptor. Lastly, a very important function of antibodies is something called antibody opsonization, which enables phagocytosis of whole extracellular pathogens such as bacteria and fungi. We've heard about opsonization before in the context of complement signaling, where the pathogen's surface is decorated with C3b particles that bind to CR1 that's expressed by phagocytes, and this is responsible for triggering phagocytosis. Antibody opsonization is just another mechanism for decorating a pathogen surface with a marker that can be recognized by phagocytes. In this case, if circulating antibodies recognize an antigen that's expressed on the surface of a microbe, they will bind to the pathogen and coat its surface. Note that this is the same process involved in classical complement pathway initiation that we talked about a few slides ago. However, in the case of opsonization, a phagocyte, such as a macrophage or neutrophil, that expresses corresponding Fc receptors can then bind to the Fc regions of these antibodies, and this triggers receptor cross-linking and phagocyte activation. This leads to phagocytosis and then destruction of the pathogen by some of the mechanisms that we covered during our phagocyte lecture back in week 2. Now that we know more about each of these antibody isotypes and the functional effects that they have on the immune system, I want to tie this information back to the types of cytokines that stimulate class switching and how these relate to specific types of infections. For this, we can look at isotypes involved in type 1 and type 2 immunity, which are mounted against intracellular pathogens or parasites, respectively. I've listed some of the other cell types and cytokines involved in each type of immune response below this chart. Now, I've modified this table from the book, which outlines cytokines that induce class switching towards certain antibody isotypes. I've added a column on the left that lists the type of pathogen that is associated with each specific cytokine and general immune response type, and I've also added a column on the right that lists relevant immune responses that are mediated through the functions of the induced antibody isotypes. I know that this is a lot of information, but I want you to focus on how these factors are linked together in a way that really makes sense in terms of getting B cells to produce the classes of antibodies that are most useful for a certain type of infection. In the top row, we have IL-4, which is a type 2 cytokine produced in response to parasitic infections. IL-4 detection in germinal center B cells triggers class switching towards IgE and IgG isotypes. IgE antibodies can induce mast cell sensitization through Fc epsilon receptor activation, while IgG antibodies can bind to the surface of a parasite and recruit eosinophils for targeting in a mechanism of IgG function that we didn't cover yet. These responses make sense when applied to the initiating parasitic infection, since parasites are too large to kill by methods like phagocytosis or complement signaling. Instead, the antibodies that have been specifically tailored to this type of parasitic infection function by inducing antiparasitic effector responses, and these are relayed through the appropriate cell types that express corresponding Fc receptors. In this case, type 2 cytokines trigger IgE class switching that is best at stimulating type 2 immune responses. In the bottom row, we have interferon gamma, listed as an inducing cytokine, which again is associated with type 1 immunity against intracellular pathogens, such as intracellular bacteria or viruses. Interferon gamma detection in germinal sensor B cells triggers class switching towards IgG isotypes, whose most relevant function in response to intracellular pathogens is probably NK cell killing of infected cells by ADCC. Again, this response is mediated through an antibody isotype that encodes effector functions corresponding to type 1 immune responses. If you read through this table with the information that you've learned so far in this course, it should help emphasize a couple of key facts. The first is that the effector functions encoded through certain antibody isotypes match the type of infection that initiated B cell activation and antibody secretion in the first place. In this way, isotypes allow antibodies to fine-tune the immune response towards the most appropriate effector cell types and mechanisms of antibody-mediated immune protection. The second point is that immune responses to a pathogen involve many different cell types and effector molecules of both the innate and adaptive immune system. Although these components vary with respect to their localization, the timing of their activation, their specificity, and their effector functions, they all must work together in order to coordinate a successful immune response. I think that antibody functions are an excellent way of demonstrating this, since they are optimized by T cell help, and in order for antibodies to function, most of the time they need to activate various innate immune cells and mechanisms that we covered at the beginning of this course. Much like you did at the end of the week 4 lecture on T cell function, please try to think about how to place B cells and humoral immunity in context with the other parts of the immune system that we've learned about so far in this class. I want to end this lecture by briefly going over aspects of memory B cells. Like the T cells that we learned about in our week 4 lectures, since B cells are a part of the adaptive immune system, they have the ability to form long-term immunological memory that protects the host from future reinfection with that same pathogen. If you remember back to our introductory lectures, this is a key final goal of a successful immune response. Again, as with T cells, the lymphocytes of the adaptive immune system, or the B lymphocytes of the adaptive immune system, are responsible for mediating this type of highly specific immune memory, and this is a result of their highly specific lymphocyte antigen receptors. B cell memory also requires differentiation mechanisms that yield stable pools of memory B cells, which are maintained long after the successful clearance of a given pathogen. And again, this is critical in order to protect the host from future re-encounters with that same pathogen. So let's end lecture today by quickly looking at how B cells accomplish this final goal of an immune response. At this point, we know that plasma cells are activated B cells that have proliferated and are capable of secreting large amounts of antibodies. These can consist of both IgM plasma cells, resulting from a B cell that did not form a germinal center, or isotype-switched plasma cells resulting from germinal center reactions, where they underwent affinity maturation as well as class switch recombination. Either source of plasma cell can differentiate to become a memory B cell, which can then take up residence in secondary lymphoid organs such as the spleen and lymph nodes, or primary lymphoid organs like the bone marrow. Memory B cells can be characterized by increased expression of some surface markers that are related to their function as APCs. These include markers like MHC class II as well as CD80. These memory B cells can undergo rapid reactivation in response to antigen, which explains why secondary immune responses are often characterized by higher levels of class-switched antibodies, such as IgG, that are produced more quickly than during initial infection. Memory B cells can also re-enter germinal centers to undergo additional rounds of somatic hypermutation, which again improves the quality of the antibodies produced by that memory B cell. We will revisit the concept of memory B cells as well as their reactivation in week 8 of this course when we discuss vaccine design. The importance of B cells and their antibodies towards adaptive immune memory is especially apparent when you compare the relative magnitude of responses offered through antibodies versus memory T cells. This graph shows curves of the relative responses generated through antibodies in blue, CD4 memory T cells in orange, and CD8 memory T cells in purple. And this is, each of these are in response to immunization with the smallpox vaccine. Although T cell memory is relatively long-lived, these responses do start to decay after about 15 years or so. However, the levels of circulating antibodies against smallpox that are induced through vaccination are sustained for an extremely long time following vaccination, on the order of a few decades. And keep in mind that this graph is just showing circulating antibodies, So consider that even in the absence of circulating antibodies in serum, that memory B cells that are in the bone marrow or secondary lymphoid organs could also be stimulated to produce even more antibodies upon their reactivation. We observe these types of responses following many different vaccines or natural infections, which really highlights the importance of humoral immunity and memory B cell formation, as well as their contributions to adaptive immune memory. We will discuss the importance of memory B cells again during our Week 8 module on vaccines, so just try to keep these concepts in mind once we get to those future lectures. In summary, today we've learned that B cells producing immunoglobulin molecules of a given specificity can secrete these molecules as antibodies with any one of a number of different isotypes, and that each antibody isotype has important effects on the distribution as well as the function of those antibodies. We've established that the 4 functional isotypes of immunoglobulins differ with respect to their affinity as well as their avidity, the tissue sites where they are found, and the types of infections that they are best at defending against. We also summarized the 5 major functions of antibodies, which isotypes are best at each of these functions, and how these influence downstream immune responses. We also briefly discussed how memory B cells can confer long-term immune protection through antibody secretion, and all of this information is summarized in the bullet points listed on this slide. You are now finished with the lectures in the Week 5 B cell module for applied immunology. Please refer to the corresponding reading assignments in the Janeway textbook to help clarify topics that we've covered in lecture. You'll also have an extra credit quiz based on this material, as well as a discussion board prompt for this week. So if you want to submit a response as one of your 4 graded discussions for this course, this week's prompt asks for a summary of a B cell malignancy. And these are cancers that arise from mutations in pathways controlling B cell development, signaling, or proliferation. So if any of those topics interest you, then please take the time to submit a discussion response for this week. Good luck with all of your assignments. Next week, we will begin our Week 6 module on experimental methods that are used in immunology research. Please remember that you also have a midterm due at the end of Week 6, which will cover all of the lecture material in this course from Lecture 1B through this lecture, which is 5C. For more information, please see relevant course announcements related to the midterm that are posted on Canvas.